Yeah, so I'm David Chan. I'm a professor of biology at Caltech. And I'm also Dean of Graduate Studies at Caltech. So I've been at Caltech, well, since 2000, and I've been doing biological research, since the, 1990s, I guess. And so my lab works on different aspects of mitochondrial biology, and we're particularly interested in like how the cell maintains the quality of mitochondria. And it turns out that some of the dynamic processes of mitochondria are very important for this. So we're interested in how, you know, there are hundreds of mitochondria in a cell and how they interact with each other and fuse and divide, and how the cell decides, which mitochondria have become dysfunctional and then gets rid of them.
Yeah, that's so cool. And then how did you get interested in research in the first place?
Well, I mean, I've always been interested in human biology, and I think during college, in my sophomore year, I started taking some molecular biology courses and just found the experiments that were done and the topics that I studied to be really interesting. And so I wanted to participate in biological research. Actually, at the time, I was also interested in becoming a physician. After college, I went into an MD PhD program.
Yeah, that's awesome. And then I know that you've kind of already discussed what your research is, but what were some of your most significant findings or publications?
Let's see. So I guess when I was a postdoc, this was at MIT at the Whitehead, I did structural studies on one of the glycoproteins. So these are envelope proteins of the HIV virus, which is the virus that causes AIDS. I was interested in the problem of how HIV virus enters a cell. Like, how does it infect a human cell and enter into it? And it turns out that it's because this glycoprotein causes fusion of the viral membrane with the human cell membrane. And so I did structural studies that were very revealing in terms of understanding that process, like how this protein causes the two membranes to merge. And so when you merge the viral membrane with the cell membrane, it basically allows the contents of the virus to enter a cell. So that was one thing. And then since I've been at Caltech, we've done a bunch of studies understanding the physiological functions of mitochondrial fusion. So it's another case of membrane fusion. And so we've found that these fusion processes are very important for maintaining the function of mitochondria, and that when they're defective, they're associated with some neurodegenerative diseases.
Yeah, that's so interesting. And then is there one specific cell type that you're looking at, or is it just more general?
It's not specific to a cell type. One of the model systems that we use is mouse. And so, we study it in the mouse as an organism, but then we also study it, in cell culture in, mouse cells. But oftentimes we're also using human cells. I would say just broadly, it's mammalian cell biology.
Yeah, okay. And then what do you enjoy most about your research?
It's just the challenge of trying to figure out something interesting and new. And so, like in any scientific problem, there's what you know, and then there are all the interesting questions that we can't answer yet. And the challenge is just trying to figure out what those answers are. And it's very challenging because, they're unknown because they're often hard to study. And so the challenge is trying to figure out a way to get those answers. And so in order to do that, I work with postdocs and graduate students. And so training like younger scientists to try and solve these problems. And so it's a complicated mixture of things that I have to do, right? Like I have to train people so that they learn how to do science. We have to try to come up jointly with ways to solve these problems. And so when it all comes together and you find something interesting, it's pretty gratifying.
Yeah, okay. And then I know you just mentioned a few obstacles that you worked through in research, but what were some of the biggest challenges that you faced and how did you overcome them?
I mean, there's a lot of things you have to kind of overcome in order to, let's say, do science like at a university, right? First, you have to be able to get usually federal funding for research. So you have to be able to explain a problem and, show why it's a compelling problem, why you have an approach that might lead to giving more information about that problem. So you have to be able to get funding. Then you have to be able to like recruit young scientists to work on the problem. And then oftentimes there are some technical problems. Many of the problems are unsolved because kind of the techniques that are currently used, they don't easily result in figuring out the problem. And so you have to do it in a different way, or you have to develop new techniques. And, sometimes there are multiple techniques and it all has to come together in order for that problem to be solvable.
Yeah, okay. And then which person or mentor in your field has made the biggest impact on you?
I would say it's for most people, it would be their graduate advisor and their postdoc advisor. And for me, it's the same. So, it's very important who you choose to be your advisor because both graduate school and postdoctoral studies are kind of critical in the career of a young scientist. And, for me, both of those people were very supportive, very intelligent people, and they created an environment that I could learn in and do well in.
Yeah, that's awesome. And then were there any like pivotal moments in your research career that made you change topics or want to pursue a topic? So kind of like what led you to look at mitochondria specifically?
Yeah, so actually that was a large pivot. So I mentioned that in my postdoc, I was working on an HIV envelope protein that is involved in the fusion of the viral membrane with the cell membrane. And so then when I was applying for independent positions at universities as an assistant professor, I decided to switch topics and, apply some of the things I had learned into mitochondrial dynamics, right? And so that was a big change. And the reason I switched was in part is because when you're starting your own laboratory, it's important to pick a problem that isn't too closely associated with someone else. And so, like my previous advisor was still going to work on HIV membrane fusion. So I didn't necessarily want to keep working on the same thing. So I wanted to do a different project. And at the time, mitochondrial dynamics was kind of just starting to be appreciated. There were lots of open questions and from many different angles, like what is the purpose of mitochondria being dynamic? Is it important for the function of mitochondria? And then just even, in more mechanistic detail, like how does it happen? What are the molecules involved? How do they mediate these types of events? None of that was known. So it was like a problem where there were multiple aspects that could be studied.
Yeah, that's really interesting. And then where do you see your research field expanding to? So like what are some interesting topics that you think can be further explored within the next like 5 to 10 years?
Yeah, I mean, one topic is something that we call mitochondrial quality control. So, like, how does a cell identify? So, you have a cell, you have hundreds of mitochondria. If there are mitochondria that are defective, you have to identify them and basically throw them away. And how does a cell recognize, certain mitochondria as dysfunctional, and how does it get rid of them, right? It turns out that this problem is actually very important for the function of mitochondria. And also, there are some human diseases that are clearly result from a defect in this process. So, like Parkinson's disease, right? It's a neurodegenerative disease. There are certain types of Parkinson's disease where the defect is in genes that are involved in this type of mitochondrial quality control. So the idea is that when neurons in these patients are unable to maintain the quality of their mitochondria, they just keep accumulating mitochondria that are dysfunctional, and that causes dysfunction of neurons, right? So it has, biomedical implications.
Yeah, that's really cool. I learned a little bit about that in a class where I was learning about Alzheimer's.
Actually, multiple neurodegenerative diseases where you can often find in these diseases that the mitochondria are not functioning as well. The problem is that, the question is, what's the primary defect? If there's a disease and you have mitochondrial dysfunction, that doesn't necessarily mean that that's like the actual first defect, because there are many other defects that can secondarily lead to mitochondrial defects, right? And so that's always the challenge with studying diseases. Like, what is the primary defect? In the case of Parkinson's disease, because we have specific types of Parkinson's disease that are caused by inherited mutations. And those mutations are in genes that are directly involved in mitochondrial quality control. We're just more confident that that's a primary defect.
Oh, okay. And then kind of moving on to your journey through research, like where did you start? So did you have any basic lab positions or internships?
So I didn't really get interested in research until I was a sophomore in college. And then once I started taking classes and was interested in participating in research, because I found that some of the classic experiments that were done in molecular biology were really interesting. So then that's when I started. I started working in labs as an undergraduate.
And then I know that you mentioned how you were, I think, at MIT for your postdoc, but like what kind of different institutions have you worked at and how were those experiences?
Yeah, so as an undergraduate, I was at Harvard College, and I worked in labs over there. Then I did an MD PhD, which is medical school plus graduate school, and that was at Harvard Medical School. And then for my postdoc, I went to MIT Whitehead. And yeah, they're all different institutions. And generally, at least in science, the general advice is that you should not stay at the same institute and definitely not in the same department. The idea is that departments are relatively small and there could be kind of like an overall way of thinking about science, and you just want to experience different ways of thinking about science. And so it's better, kind of more broadening. College should be different from, grad school should be different than postdoc. And just having the different types of science environments helps you to grow as a scientist.
Yeah, that's really cool. I never knew that. And then these are questions, I guess, more geared towards high schoolers, but how did you really like find your passion for research and how would you suggest a high schooler try to find like their own interests in research?
High school students will get interested from the science classes that they learn, and then often that leads to like just some independent reading. And so many high school students, that end up going into, STEM oriented colleges, they have developed their own interests in science. And, you can also like sometimes you have some research experiences even as a high school student. So I think it's just driven by finding topics that you're passionate about.
Yeah, okay. And then what kind of skills do you think a high schooler could develop even if they don't have access to a wet lab or any like materials that you guys have?
I think it's just learning to think critically. Oftentimes, as a high school student, even if you get a lab opportunity, it's kind of hard to, be all that independent, right? Because there's just so much background that you need beforehand. But like just being exposed to labs and learning about the kind of things that laboratories do is already very helpful. And then just reading broadly about that research topic can tell you if this is really something that you're interested in or not. Because I think ultimately, if you go into science, it's going to be largely driven by your own interests in the topics, because there are lots of easier things to do. There are careers that are easier, that pay more. And so the people who stay in science are people who are just really passionate about learning more about these topics.
Yeah, okay. And then what traits do you think define a great researcher in their field? And what type of character traits would you look for in a potential lab member or like collaborator?
There are lots of traits. One is clear thinking and rigorous thinking. Like someone who really is driven by, learning and getting information and getting new ideas from data as opposed to like having a preconceived notion about how things work, right? Like you really want someone who is able to come up with ideas just, based on data. And then, of course, you definitely need someone who's honest. So, like integrity in science is really important, not over-interpreting, but making sure that your conclusions are really kind of solidly based on data. And then someone who is self-motivated, that's really important because, again, the people who end up going deep into academic science, they're people who are you basically, you don't have to tell them what to do, right? They're driven to do these things and they spend a lot of time because this is what's interesting to them. Because you can't kind of teach people to be interested in something. It's kind of a innate interest that good scientists have.
Yeah, okay. And then I'm sure that you get a lot of emails from high schoolers or like undergraduates asking to join your lab, but how do you select someone from the crowd? So like what makes someone stand out to you?
Yeah, I mean, it is a little unusual for high school students to get the opportunity to work at like university laboratories. I would say usually, if someone just gives a generic request for, working in a lab, it's not going to stand out, as compared to any other requests, right? The ones that one would pay more attention to is someone who actually has like read some of the work that someone has done and is like specifically interested in a particular project. Someone who has clearly, taken some time to figure out what the lab is doing and shows a specific interest in it, as opposed to, someone who's just looking for, any opportunity to join a lab.
Okay, so really just demonstrated interest.
Yeah. Yeah.
Okay. And then just for our final question today, what kind of tips would you give to a high schooler who wants to like go into research in biology?
I would say that the people who do the best in research are the people who are innately very fascinated by the problems that they're studying. Because when you think about how long it takes to like be independent, right? Like, for example, you go to college, then you go to grad school. Grad school could be anywhere from 5 to 7, 8 years. And then you do a postdoc, which could be anywhere from 4 to 7 years. So it takes a long time. And it's the same thing with medical students who want to do research, who do MD, PhDs, right? And so you can't think of it as going into this very long training path so that at the end of it, I'm going to be like an independent scientist. You have to be interested enough in the problems that like at every step of the way, it's actually very exciting. You're learning all sorts of new things. And so it's not about getting to the end stage, it's about like learning throughout that process and satisfying your curiosity. It's absolutely critical. Like if that's not the present, then it's a difficult, long process, and it's probably not the right career path.
Okay, so it's really like that interest in science that keeps you going.
Yeah.
So that's all the questions that I have for today, and thank you so
Okay.
much for meeting with me.